Is the screen on?
It's on HDMI. Not.
The stream's actually playing on fire. It's on.
I know the projectors are on. I don't know what's. I have my own HDMI. You want to try a different HDMI? And I look at what they did and start doing. I think because they.
We're gonna have to move the computer over here.
They're heating up. Come on. Right.
I just could be done like that. I think that's the one that I write it for 5 hours. Then I wait for
These are old machines.
and chemistry. I'm not even doing it for half of the.
That's so awesome.
Ain't it?
Dude, it's so stupid. Okay, we're gonna have to, the projectors don't want to work. We're gonna send us slides out. So if you guys could,
Right, there's a bunch of paper over there.
right here, it goes down. Here. You can try it for some.
Oh, it's flickering. I think it's gonna work. All right, sweet. All right, we're gonna get going. Sorry about the delay. Burke ran a little bit late. But that's no excuse. So we are sorry that we're gonna start this late. I know it's already late, but we'll get through the PowerPoint and then we'll have time for questions on the problem set. Could be content questions, anything. I'm gonna go through topics 1 and 2. John's gonna do 3 and 4. If at any point you have a question or you want to clarify, or I might have made a mistake, raise your hand and we're happy to work with that. So yeah. All right, so we're going to start with intro to transcriptional regulation. So for prokaryotes, the RNA polymerases are sigma factors. These are transcription factors that are found in these prokaryotic cells. In eukaryotes, it's basal transcription factors. This is more of a term definition. So if you write this on your note card, you'll be golden. I don't know. We don't really define what they are, but just know that those are the types of transcription factors in each cell. And then we have regulatory transcription factors, which control the level of gene expression, how often and frequently your genes are expressed. And these are gene specific, and there are positive and negative. And you can infer that positives would enhance transcription, so you'd be doing transcription more, whereas negative are the opposite. So sigma 70, this is an important name to remember. It helps RNA polymerase bind. And it has a consensus sequence, which consensus sequences are the most common nucleotides at each position. So in this figure, it's from Khan Academy, I believe. You can see there's different sequences, and the most common letter is T and A. So there's two that could be here. That's a bad example. But for example, on the right figure, there's A, C, A, G, A. So the consensus sequence there would be A. And what's nice about consensus sequences is they aren't totally specific. So you can have a few nucleotides wrong and still be part of that consensus sequence. So it's more of a general. So operon operations, it's a series of genes that's transcribed at the same time, and it's controlled by one promoter, and it's the same promoter. So once that promoter says go, every gene will be transcribed. Okay, the TRP operon, this is going to take a little longer. This is about transcriptional regulation. So just to go through the operon again, the promoter is where the RNA polymerase binds. It's marked with a P here with the purple blob on it. The operator is where the repressor binds, which kind of stops that promoter from binding, which is this pink blob in the bottom left, and it's on the O region. Inducible operons are usually off and they get turned on. So usually the repressor is bound in an inducible. You induce the operon to activate. Whereas repressible is usually on, and it's in this top figure, where there's no repressor bound, and you have to put the repressor there to turn it off. And that is, the repressible is what we're going to be talking about with the TRP operon. Okay, so the TRP operon, when you have low TRP levels, TRP is an amino acid, similar, you know, guys have the amino acid chart. It's just the chemical that's found in your body. And the TRP operon is how we make that chemical in our bodies and how we do our chemical functions. So when we have low levels of TRP, there's no access to TRP for proteins to be made, and thus the cell needs to make more. And so then what happens is the TRP genes are turned on, right? Because we need more of it for the cell. When there are high levels of TRP, the cell really doesn't need to make more. So these TRP genes are turned off because there's an abundant access of TRP. This is another figure of the operon. I don't know if this is the one Laura uses in class, but this is the one I had on my note card when I took the class. It worked better for me. And then what's kind of magnified here is the attenuator sequence, which is going to be the next part of this review session. So attenuation is early transcription termination. It's kind of like a police car pulling you over. I don't know if that was the best analogy, but it stops it before transcription happens too long. So as you can see on the right, the TRP genes, I'll go back a few slides. The TRP genes are these blue ones, and this attenuator sequence is right before. So it's kind of the final stage before all the genes are transcribed. Oh, sorry. And there are 3 hairpin loops that form. The 1 and 2, which is a pause, and it doesn't indicate anything about if transcription is going to stop or continue. There's 2, which is the anti-termination or the go signal, and then the genes are transcribed. And then the 3 and the 4, which signifies termination. And we'll go through these, and I'll have a slide that shows a good overall view. All right, so back to when we have high levels of tryptophan. This repressor is only active when tryptophan is bound. So if we have high levels in the cell, there's lots of these little yellow particles. They're going to bind to the repressor, and the repressor is going to turn on, and it's going to bind to the operator region, and no transcription will occur. This is probably the most simple because the attenuator is not happening. So if you have high levels of TRP, the repressor is going to be bound. Nothing's going to happen.
Can I say something?
Yeah.
Very, very, very common for Laura to get something on the exam saying, in high levels of TRP, but a mutated repressor that doesn't work, what happens? So think about scenarios that just off of that could be, because now you're used to Laura questions from experience.
Traumatic experience.
Not traumatic, Andrew, but think about ways that Laura can modify like the normal wild type functional version of the process, and think about how she can modify that because you're a little bit more used to like what the questions are supposed to look like.
During low levels of TRP, right, the cell really needs to make this TRP, this amino acid that's crucial in protein generation. So what happens? Oh God, the picture is terrible. That there's full transcription and full translation, and all the genes are transcribed. There's nothing to do with the attenuator. Everything goes swell in the cell when there is low levels of TRP. Now, the attenuation mechanism. So this is, again, this is right here, right after the operator and before the genes are transcribed, we have this attenuation sequence happen. And so the mechanism, regardless, yeah, you go ahead. The genes get transcribed, or sorry, everything goes swell. Sorry, yeah, sorry. Yeah, I must have. If with high TRP, if the cell can detect all this TRP in the environment, the repressor, this pink guy, will bind to those TRP molecules and come back to the operator and say, nope, we're not transcribing. So this stops it right at the brim. It's like before transcription can even start, right? This RNA polymerase, which does a great job of transcribing, it can't work if the operator is bound. Yeah, so this mechanism, regardless of if there are low or high, this first 1 and 2 is going to happen, this first hairpin loop. So in prokaryotes, not like eukaryotes, transcription and translation, so transcription being DNA to RNA, and then translation being RNA to protein, can happen simultaneously, right? Does anyone know why that can happen? Any guesses? Yeah. No nucleus, perfect. Yeah, everything's shown in the cytosol. So when it starts transcription, it will start translation as well. It makes it a lot more efficient, and the cell loves efficient processes. So what happens is transcription will occur. This is the operator. It'll go through the attenuator, right? And it'll get to right here, and it will stop. It will take a pause because this hairpin loop will form. Hairpin loops are just nucleotides. You know how an RNA strand is single-stranded? It's basically zippering together, right? And this can be broken, but it's an intermediate interaction. And this gives a chance for this ribosome, which is doing the translation part. So it's taking this mRNA strand that this RNA polymerase is making, and it's turning it into a protein. It is tricky, but these are working simultaneously, as we mentioned. So this hairpin loop, this 1 and 2, really give it a pause and let the ribosome catch up so that it's not too far behind. On the other hand, in 2, on this top right part, the ribosome will get stuck in region 1. When would it get stuck? So UGG, those are 2 codons that code both for tryptophan, right? So if the cell has low levels of tryptophan, this ribosome is going to be reading this and saying, okay, I'm waiting for a tRNA of tryptophan to come in. No tryptophan is going to come in, right? The cell doesn't have any. So what it does is it gets stuck. It can't move past this codon. This codon signifies tryptophan. There's no tryptophan in the cell, right? So it'll get stuck. It can no longer produce a protein. What happens then is that this RNA polymerase, it doesn't care. It's just taking this DNA and making mRNA. It'll keep going. And what happens is a zipper forms. This is the 2-3 loop, which results in termination of no termination, sorry. So this is very tricky. So please stop me if you want to clarify anything. There's no tryptophan. UGG codes for tryptophan. This ribosome's looking for a tryptophan tRNA. It cannot find it. This RNA polymerase doesn't care. It'll keep going, and this 3-4 hairpin or 2-3 hairpin forms. And that signals to the cell. What would that signal to the cell? It signals that the cell does not have tryptophan. This is a glaring signal to the cell that we need this tryptophan. Because this ribosome gets stuck, we need the tryptophan to be made. On the other hand, what if we did have tryptophan? What if instead of getting stuck, this guy found two tryptophan tRNAs and it just keeps going through, right? So it goes past here, and then the 3 and 4 hairpin. Why did I write 2, 3? This is just for the 2. Oh, this is just for 2 and 3. Okay, the next slide will have it. So what happens is this 2-3 hairpin forms, transcription and translation. Transcription can continue, translation doesn't. And this full length mRNA transcript by this orange guy will be produced, but this ribosome will be stuck on that tryptophan codon because there's no tryptophan. On the other hand, this is definitely tends to be an exam question. What if we initially had low levels of tryptophan? So this tryptophan started, right? There's no repressor bound. This promoter, the RNA polymerase binds and it starts going. But then what happens when we get to high levels of tryptophan? Let's say the cell, by phagocytosis, the cell gets a lot of tryptophan into its cytosol, and it no longer needs to produce tryptophan, right? It has its abundant quantity. Dude, what? My slide got deleted. Okay, there's a bunch of YouTube videos, but they all stop. No. Okay. Okay, so what would happen in that scenario is a 3 and a 4 hairpin would form. Instead of getting, it's going to start the YouTube video, I swear to God. Okay. What if there's tryptophan? Does the phagocytosis happen? 2 tryptophan molecules come in. This ribosome, it's chilling. It keeps going. It does not care. There's no lack of tryptophan, so it will go through that process. What will happen is there's this 3 and 4 hairpin will form, and that signifies to the cell that there is tryptophan available, and we do not need to produce more tryptophan. And so what will happen is this thing called attenuation, where if the RNA polymerase, this is our RNA polymerase, right? It's going. The 3-4 hairpin loop forms. If this is our RNA strand, oh God, I feel bad. Okay, this black part is our mRNA, okay? This is our RNA polymerase. It's going. The 3-4 hairpin loop forms, bang. The mRNA gets shot out of the RNA polymerase, so no more RNA polymerase function. And the mRNA strand is just dead from there. Yeah, and then it gets destroyed. So again, it's, here, I'll put the, right, this orange guy, it's chilling, it's going, it's producing this mRNA. Once that 3-4 hairpin forms, bang, it gets shot out. I think there's a few animations. Laura sent one this afternoon, but you can see it in the cell. Basically, this mRNA transcript just gets taken out, and that is the end of the RNA polymerase function, and that's attenuation, which means no more transcription, no more translation. And this only happens when you start with low levels of tryptophan. So if we go back to here, right, we started here, so there's no repressor bound to the operator. RNA polymerase is like, okay, I got this. It starts going, and then once that 3-4 hairpin loop forms, it just gets taken out. Yeah. It's similar to the 2 and 3 and the 1 and 2. It's a great question. It's like a zipper. So these nucleotides, right, the A and the T and the G and the C, they will find each other in these hairpins, and they will form this interaction. And it's a temporary interaction, right? Because it needs to be broken, but it signifies things to the cell. So it's nucleotide base pairing, but it's only temporary. It's like a signal or yeah, go ahead.
It happens. This is also Laura's topic notes, but RNA, unlike DNA, can be complementary on the same strand.
Yeah. So it's like,
so the 4 regions on the operator. That's marvelous.
thank you. I'm actually an artist.
Yeah, if you look at the slides, like for the reported read lectures or whatever, like when the 1 and 2 forms, the 3 and 4 automatically forms right under. But then if only the 2 and 3 form, what really happens is the ribosome senses it. Because there's no attenuation, the polymerase keeps going. It breaks that hair or that hairpin, and it just keeps going. Right?
Oh, sorry, I wasn't listening. I was
breaks the 2 and 3.
Yes.
Yeah, the ribosome. So when attenuation does, or when anti-termination happens, which is the attenuation of 2 and 3, the ribosome keeps going and breaks that. It's like massive. It's like a really big structure, and it just completely overruns. 2 and 3 is when there's just.
Yeah, so if it's stuck in low levels, right, this pause will happen where this hairpin is brilliantly drawn by myself. It'll form these complementary and that it's a signal to the cell. These hairpins are just signals. This signals, okay, we're gonna wait for this ribosome to catch up. After this happens, it goes in and it continues. And then this 2 and 3 form, right? Because the ribosome gets stuck on the tryptophan codon, right? Because it's coding for tryptophan, but there's no tryptophan around. So it gets stuck here, and that's another signal to the cell saying, okay, we still don't have tryptophan, let's keep it going. And then the full length mRNA is transcribed, and then from there, the tryptophan genes get transcribed. Whereas
Well, essentially, you're not really out of tryptophan, the amino acid per se. You're out of tryptophan tRNA. There isn't the RNA molecule bringing the tryptophan for the ribosome to synthesize it.
when did we talk about doing that? That's later. Did you guys
Yeah,
talk
well, we
about
did- we
tRNAs
did talk
in
about the
Bio
different types of
151? So it's a similar mechanism here. When it sits on that codon, it's waiting for a tRNA to come and plop the amino acid in and then leave. But if there's no amino acid, it's just going to sit there. And then that's a signal to the cell saying, okay, we really need tryptophan, right? We don't have it. And the previous page had a lot of YouTube videos. I hope they don't all play. This is one of the trickier concepts, and this will definitely be a big part of the exam. So if you can write down this on your note card and you have a good grasp of this process, you will be golden for this one. And as John mentioned, Laura will frame it as your repressor doesn't work anymore, or
Your operator region,
the operator region is no longer bound, and you have there's like
low
plenty of
level, or you have high levels, but your repressor has no affinity for tryptophan. What happens if you have high levels of of tryptophan, but your repressor has low affinity for tryptophan because of a mutation? What would happen? So it's still synthesized.
Yeah.
Whereas if they ask the opposite, if there's none but your repressor has high affinity for your operator, it's and the cells will die.
Yeah. And your problem set, as you've probably gauged, is a really good reference in terms of what kind of questions the exam will have. So I'm sure there's a question on the TRP operon. So if you can go through that and you can, manipulate the questions just a little bit, either add an inactivation to an activation, and answer that, you will be in a good spot. And with that, I think it's
All right. Non-coding RNAs. So, non-coding RNAs are RNA molecules that don't code for proteins. When we say RNA without like any prefix, your mind usually goes to messenger RNA and mRNA. That's the one that's being translated by the ribosomes. But there are different RNAs that do not code for proteins. And so there are different roles and functions within the cell. The two major ones that we cover are reaction catalysis, because the ribosome has an enzymatic part, which is actually what's translated. So it's the ribozyme, and then also gene expression regulation. So ribosomal RNA, just talked about it, catalytic part of ribosomes. And then transfer RNA is another common one that I actually just mentioned while Van was talking about the trip codon or the trip operon. And it brings amino acids to the ribosome during translation. And it's complementary to the mRNA strand. So then these are also some of the other RNAs that Laura mentioned in her topic notes and in the videos. SRP we mentioned on the previous topics. CRISPR is very prominent nowadays because of research. And then long non-coding, small nuclear RNAs are very specific. You probably won't be asked about it. You just need to know they exist. So this leads us into the bigger part of the RNA section of this topic, which is RNA interference. Silly meme, but gene silencing is what happens because of RNA interference. So RNA interference is an eukaryotic mechanism of gene expression regulation, at least for this class's purposes. Prokaryotic cells have similar mechanisms, we just do not cover them. And specifically, we'll be talking about post-transcriptional control of mRNA via suppression of the gene that the mRNA is coding for. So then here are the RNAs involved. We have the RNA-induced silencing complex RISC. It is quote unquote an RNA interference machine. It just goes in through the pathway that we'll talk about in a second, and it silences the genes. And then we also have short interfering RNA, siRNA, which we will also be talking about, along with microRNA or miRNA, short. So RISC, again, is an interference machine complex. So there are multiple different proteins involved. And the general mechanism is that RNA is going to bind to RISC, and then that RNA that is bound to RISC is going to be used as a complementary binding strand. Basically, what Laura did when we were like in class when me and Ben were students, she like opened her arms and basically said, "Oh, there's like a complementary strand of RNA right here. It's just going to go around the cell trying to like find the mRNA that it's not there." So that helps you. That helps you. That doesn't help you. It's just looking for specific RNAs that it can complementary bind to. And it regulates translation or degradation of mRNA depending on the mechanism that is followed. And there are two pathways for this that are using RISC, and it's the miRNA pathway and the siRNA pathway. And we will look at them right now. So miRNA specifically is a post-transcriptional regulator. It functions as one of the guide strands for RISC, and it regulates stability and localization of RNA. siRNA is different on this aspect. And then it's small 20 to 25 base pairs of dsRNA, which is another kind of RNA. And very key for miRNA versus siRNA. I did a comparison chart afterwards, but very key, it is endogenously expressed. What does that mean? It's encoded within our genome. It is made in our cell. And it is expressed in the nucleus. Though it is not incredibly prevalent in humans, we still have it. So the pathway. So originally the polymerase is going to transcribe the initial transcript. And so the PRI miRNA is created. Laura mentions this in one of the topic videos. That chart says pre-miRNA on it. It's supposed to be PRI. Sorry. And then, so once the PRI miRNA is created, you have the steps of cutting. In this chart, Drosha is just a pair of scissors. In reality, Drosha is not a pair of scissors, but essentially it is cutting the different miRNAs that are still premature in the nucleus. And once that processing is done, the now pre-miRNA, because now it is a pre-miRNA, gets exported from the nucleus to the cytosol. And then continuing, second cuts. So then the miRNA that is already in the cytosol is going to get further processed by Dicer, and it's going to get split apart. So it won't be like double-stranded anymore. It will be single-stranded. And now it's ready to bind to RISC. RISC is then going to unwind and choose one of the strands because there's two for each pre-miRNA. And now it's going to, again, choose one of the two, and it is going to either modulate how translation and degradation is going to go, or how sequestration into the P bodies is going to go. So if you have perfect base pairing, then the mRNA that gets bound to, that RISC binds to, is going to get degraded by Argonaut. And I'll talk about this on the next slide. If it is imperfect, like on the drawing, there's a little gap on the binding, then it gets sequestered into the P bodies. And Laura mentioned that in the video, nobody really understands what goes on in the P body yet. Like people just know that it goes into the P body and it disappears. But no one knows yet. So right there, yeah, I guess I skipped a step by saying it, but so miRNA in humans usually don't exactly base pair. Usually a lot of what's happening is just sequestering into the P bodies. The mRNA degradation happens in other cases in different ways. But so perfect base pairing gets degraded, imperfect, it gets sequestered. And this is very key, especially for like larger cells and like larger multi cellular organisms. What this allows for in imperfect binding is it allows that singular like strand to control a variety of different like mRNAs. It doesn't have to be base pair specific like 100%, so it's not degrading it per se, but you're able to like match a whole bunch of different mRNA sequences with just that non-specific binding. Because it's not matching 100%, and that just becomes more effective, like Laura was talking about in class. Cells have resources, they don't want to overuse resources, they want to, optimize the amount of stuff that they use. And then the more complex part of the exact matches, if it exactly matches, then Argonaut activates, and then that's the enzyme that leads to degradation of the mRNAs. Any questions? No? Okay. So then, second part, siRNAs. So, like miRNAs, they are post-transcriptional regulators, and they also act as guide strands for risk. However, it does not control localization for miRNAs. It is only for stability. And again, it is small, 20 to 25 base pair of dsRNA. This one, however, is exogenously expressed. What does that mean? Exo, from outside of the cell. It's usually coming from like a viral particle. Most cases. And this can protect organisms against foreign invaders. That can contain that specific dsRNA. It's kind of like a similar process to how CRISPR works, but CRISPR is different. And then usually for siRNA, it is an exact base pair that is going to match. So this is the pathway. It starts in the cytoplasm because it is not in the nucleus. It is exogenous. It's just going to go into the cell. And so the dsRNA or the shRNA is going to be cleaved by dicer. Upon that happening, you have an siRNA duplex, which is just a processed dsRNA. And then RIS is going to go in and unwind that, and then pick one of the two strands to act as the guide strand. Again, they're complementary, so they basically serve the same purpose, but it's for mRNAs that have the exact opposite strand. And so by doing that, what you basically do is you double the amount of controlling that you can do with one strand. And so you're going to look at the diagram. I also numbered it by step to match. But so upon that, the RISC siRNA complex is going to form. And then it's going to complementary base pair with the mRNA. Once you have an exact base pairing match, the RNA is going to be cleaved or nicked by Argonaut 2 subunit of RISC. Again, the process is pretty similar. What's going to change really is where the source is coming from. mRNA is coming from the nucleus, siRNA is coming from something else. So it starts in the cytosol already. It completely skips the getting exported out of the nucleus step. And this process with siRNA leads to degradation or silencing. Most of the time, not 100% of the time, it is exact base pairing. And that's the little chart I was telling you guys about. This is a drawing of like the two pathways side by side if you want to compare it. This is something that's probably useful as part of your note card. It just really evens it out. They're not really that different, besides the fundamental aspect of where the source is coming from. But yeah, so there's like a drawing of the pathway if you're a visual learner, and they are similar yet different in different aspects. But this specific part is less complex, I would say. Like it demands a little bit less time than like actually fully understanding the trip operon stuff. So yeah. And I think that's it for this. So feel free to email us with any questions. And yeah, we're gonna stick around a little bit for questions. And
Yeah, does anyone have any problems with that question or want us to go back to a content type question?